Hello, Dr. Namira, madam.
Yes, sir.
Uh, Welcome you, madam. After a long time, I think
Yes,
you
sir.
was not
After a
well.
long time.
Uh,
I was not well,
yeah.
but
So
now I'm.
now it is okay, madam. You can continue now.
Yeah, yes, sir.
Yeah.
I'm
Please
fine now.
proceed, madam. Please
All
proceed.
right. Okay. So, hello everyone. Welcome to Tawheed Foundation at Allied Healthcare Sciences, Bangalore. So, today's class is for second year BSc MLT students, Parasitology. So, parasitology is a discipline that studies the structure and physiology of parasites, their host, and host parasite interactions. So, in biology, parasitology studies the structure, physiology, and interaction of parasites with their host, but in biology, parasites are not determined by the nature of the organisms or the environment, but these are classified by their mode of life. Although parasitology originated in the zoologic science, it is today an independent interdisciplinary field highly influenced by microbiology, immunology, biochemistry, and other life sciences. So, traditional medical parasitological studies have included studies related to three major groups of animals: parasitic protozoa, helminthes, and arthropods that directly cause disease or act as vectors of various pathogens. A large number of infections of human are caused by parasites which range from relatively benign to fatal, with some parasitic diseases being the cause for major human health problems throughout the world. So it ranges from mild infections to life-threatening diseases. Traditionally, parasitology has been limited to the identification of parasites, their mechanisms, and diseases causes in plants, animals, and humans. So, modern parasitology research is more focused on the determination of signal transduction mechanisms controlling the behavior, survival, virulence, and gene expression of parasites, which have a crucial role in influencing the outcome of the interaction between host and parasite. So, parasitology has importance in veterinary and ecology, where the diseases caused by parasites in animals and the role of parasites in the ecosystem are the two major areas of focus. So, most parasites, unlike viruses, are not host specific and can infect multiple hosts. These depend entirely on the living being for their nutrients and survival. Parasites can be differentiated from fungi based on their host, as parasites rely on living beings while fungi depend on dead particles for survival. Parasites are mostly macroscopic and belong to the invertebrate subphylum of the animal kingdom. Diagnosis of parasite diseases is based on the identification of infectious agent, which is mostly made through microscopic examination. With the continuous emergence of new parasites and parasitic diseases, parasitology is considered a necessary scientific discipline. Types of host. So first, what is host? Host, it is a reservoir which is an intermediate host where the parasites develop and from which infections can be acquired by other hosts. For example, host consists of echinococciasis. The host may be of the following types. First is definitive host, second is intermediate host, third is a reservoir host, fourth is paratenic host, and last is vector. We'll go one by one. First, we'll start with definitive host. So, the host in which the adult parasites reproduce sexually, for example, Anopheles species, is referred as the definitive host. The host could be human or non-human living beings. Example, anopheles, mosquito for plasmodium. Second is intermediate host. So the host in which the parasite undergoes asexual multiplication is known as an intermediate host. For example, life cycle of malaria parasite, humans are intermediate host. So a few parasites require an intermediate host to complete their life cycle. Some parasites need two intermediate hosts where the various larval stages of the parasite complete their development. These are referred to as the first and second intermediate hosts respectively. Third is reservoir host, the intermediate host in which the adult form of parasite lives but cannot grow and develop further, and which is not necessary for the parasite's life cycle, is called paratenic host. Reservoir host, a host to which the parasite is available for transmission to another host and it does not get infected by the disease commonly. Example is dogs in echinococcisis. Paratenic host, the intermediate host in which the adult form of parasite lives but cannot grow and develop further, and which is not necessary for the parasite's life cycle, is called paratenic host. So freshwater prawns for Angiostynococcus cantonensis, a big suitable fish for larvae of these include in paratenic host. Last is vector. A biological vector is an organism capable of transmitting the disease agent or the causative organism from infected host to other host. Example is sandfly for Leishmania species. Parasitic life cycle. It is of two types. First is direct, or you can tell simple life cycle. Second is indirect or complex life cycle. So the life cycle of the parasite starts from the point of entering the host and ends with existing the host. So it can either be direct simple life cycle when only one host is affected, or the mode is indirect, that is complex one, which means involving one or more secondary hosts. So we'll start with direct simple life cycle. So here the parasites can develop in a single host. Its life cycle is considered to be direct simple. Example is Giardia, Lamblia and Ascaris lumbricoides. Second is indirect complex. So when a parasite requires two hosts, one is definitive host and another is intermediate host to complete its development, it is referred to as indirect life cycle. Example is Plasmodium leishmania species, Taenia solium, Taenia saginata, etc. So life cycle knowledge is vital for control as well as prevention. Host parasite interaction. There are four types of host parasite interaction. First is symbiosis, second is commensalism, third is parasitism, fourth is mutualism. So symbiosis, the relationship between a host and a parasite can be divided into this. We'll start with symbiosis. So it is the close association competition between the host and the parasite. Both are mutually dependent on one another. One cannot exist without depending on the other. So none of them get hurt by each other in this symbiosis relation interaction. Commensalism, it is a relationship in which the parasite has everything from the host side of a profit without the host suffering any losses in the process. In another word, a commensal has the capability to lead a life on its own term. Third is parasitism. It is a relationship in which the host works to the advantage of the parasite while always being detrimental to the host. So the host does not get anything in return in parasitism. Last is mutualism. Both benefit actively in mutualism. It is simply defined as the coexistence between the host and a symbiont in that none suffers although both gain benefit from each other. Yet this may last longer or may be short term in nature of relationship. Mutualism is of species of symbiosis. One example is kind of simple where some flagellated protozoa that inhibit the gut of termites. So as the protozoa are purely and simply carbohydrate feeders, they obtain all their food from termites. So these are the four important host parasitic interaction. Classification of parasites. So it is divided into two parts. First is protozoa, second is helminthes. So protozoa kingdom is Protista. It is unicellular in nature. Helminthes kingdom is Animalia, which is multicellular. Protozoa is again further divided into four parts. It includes amoeba, flagellates, sporozoa, and ciliates. Amoeba, it is typically amoeboid and it will use pseudopodia for its movement. Example include Antamoeba histolytica, one basic common example. Second is flagellates. It have one or more flagella. It is generally found in intestine. Example is Giardia. Third is Sporozoa. It undergo complex life cycle. Example is Plasmodium. Last is Ciliate. It is a complex protozoa. Example is Ballantadium coli. Helminthes, it is further divided into three parts. First is nematodes, cystodes, and trematodes. Nematode is nothing but roundworms. It is elongated and tapered at both ends. It is unsegmented and example include Ascaris species. Cystode, it is tapeworms. It have a ribbon-like chain segments. Proglottids, example is Taenia solium. Last is trematodes, that is flukes. That is typically flattened in shape, leaf-shaped structure with two muscular sucker, cuticle, hermaphrodites. Example is Fasciola hepatica. Diagnosis of parasitic diseases. An important modality in the diagnosis of parasitic infections, the laboratory diagnosis is an important aspect of the management of disease. So, the following diagnostic techniques are used for diagnosis of parasitic infections. First is macroscopic examination. It includes visible worms or segments in stool. Second is microscopy, wet mount slide, saline, iodine staining. All this include in microscopy examination. Culture methods, it include parasitic growth in specific media. Fourth is molecular method. It include polymerase chain reaction, DNA probes. Last is immunodiagnostics. It includes ELISA and rapid test. We'll go one by one. We'll start with microscopic examination. So first, morphological identification techniques. Looking at the morphology, of the parasite, it is possible to categorize them macroscopically as well as microscopically. Of course, the different forms that various parasites can be observed at different morphological levels. So, examination of feces. Stool samples. Stool specimens should be obtained from the well, non-leaking screw cap jar with a wide mouth and should be handled gently as so not to contact infections from other organisms present in the stool. Ideally, the specimen should be taken before the commencement of antiparasitic drugs and at a time closer to clinical manifestations. So, three stool samples given at least 48 hours apart are sufficient to help establish a diagnosis of intestinal parasitic infections. So, liquid stool sample need to be processed and get ready for examination in within 15 to 30 minutes, while semi-solid ones in about 1 hour. Formed feces should be examined within 24 hours after they were collected. During storage, the trophozoite may rupture, it loses motility and may enhance appear as artifacts. So many preservatives may be used for this purpose. For instance, 10% formalin or polyvinyl alcohol to preserve the cyst and egg morphology of the parasites. Second is microscopic. It present in acute amoebic dysentery, intestinal schistosomiasis, and invasive ballantidiasis. So the dark red color of the stool means upper GIT bleeding, while the bright red stool indicate bleeding from lower GIT. Non-bloody pale offense frothy stool with fat formed in small intestine in case of giardiasis. This may contain roundworms, threadworms, or segments of tapeworms in adults as well. Microscopically, here saline is dropped and Lugol's iodine is put into different corners of one slide. A small amount of feces is manually stirred using a stick so that it becomes a homogeneous smooth liquid suspension. A cover slip will placed on the mount and observed at low power of lens, that is 10x, or high power of lens, that is 40x. So wet mount slide will be prepared and observed under microscope for parasitic infection. Molecular methods. There are two types of molecular methods for the identification of parasites. They are DNA probe method and polymerase chain reaction. So DNA probe is also a nucleotide sequence attached to each of its radioactive isotopes. So it will involve a portion of the parasitic DNA detectable within the clinical samples available. This is highly specific and reproducible. At present, there are protocols for the detection of specific DNA sequences using these DNA probes for parasites such as Plasmodium falciparum, Bancroft, Bancrofti, and so on. PCR-based assays involve several additional steps after the choice of primers and probes. So real-time PCR amplification of the DNA will be done for molecular method of parasitic infection. Last is immunodiagnostic, ELISA and rapid test will be included. Anti-amoeba histolytica, it will cause amoebiasis. So anti-amoeba histolytica, many amoeba inhibit human gastrointestinal tracts such as Dientamoeba fragilis, Antamoeba histolytica, Antamoeba coli, Antamoeba disper. All these species, these amoebae are commensal and do not cause harm to the host, but some amoeba such as fragilis and histolytica are human pathogens. The infection of amoebiasis is highly developing and underdeveloped countries. It is also the third leading parasites that cause annually 1 lakh deaths around, which is due to low socioeconomic status and poor hygiene practices. So protozoan parasite infecting large intestine, it causes amoebic dysentery and liver abscesses. It infects 30 to 50 million annually in humans. This is the image of gastrointestinal amoebiasis, Antamoeba histolytica. Morphology. So, pseudopod forming anaerobic protozoan parasite, it is actively motile by crawling or gliding. You can see crawling or gliding. It exists in three forms: trophozoite, precyst, and cyst. These are the three forms. Trophozoite, it is around 10 to 50 μm. It is motile. Precyst and last is cyst, that is around 10 to 16 μm, 4 nuclei, and it is mature in size. So when immature with a single nucleus and after maturation forms 4 nuclei. Cyst can survive in soil, water, and especially moist food. It is divided by binary fission. It is sensitive to drying, heat, and chemical sterilization. At low temperatures, pseudopodia formation and motility are inhibited. The diameter size of trophozoite is around 10 to 50 μm with a single nucleus. Transmission. So the most common source of contamination is ingesting contaminated food and water by human feces. It is also transmitted indirectly through the objects or hands through anal or oral contact. At a certain level, cysts are resistant to chlorine used in drinking water, making it ineffective. Poor socioeconomic condition and sanitation are the major cause of endemic illness in developing countries such as India, Africa, and parts of Central and South America. Migrants and travelers to endemic areas are also the main cause of parasitic infection in the United States. Another common spreading cause of the disease is through asymptomatic carriers, especially food handlers. Consumption of food, crops, and aquatic animals that are irrigated with fecally contaminated water is also a route of transmission. Sexual transmission among homosexuals has also been reported for the transmission of these diseases. Pathogenesis of amoebiasis life cycle. It started with ingestion of mature cysts, then it leads to excisation in intestine, then occurs trophocyte multiplication, then encystation, and last is cyst excretion. So in detail, pathogenesis. So what happened? The infection starts within 2 hours when around 2000 to 4000 cysts are produced in the human after ingestion. Entamoeba histolytica infection disrupts the protective mucus coat and of the stomach and detaches the enterocytes. It then penetrates the lamina propria, this lamina propria, and lyses the cell, secreting pro-inflammatory molecules such as interleukin 1 beta, interleukin 6 and 8, interferon gamma, and tumor necrosis factor, and causes inflammation within 4 hours. So the virulence of this parasite is attributed to its capacity to destroy the host tissue by the secretion of various molecules. It anchored glycoconjugates present at the surface of the parasitic cells are involved in the interaction of host and parasite. The damage to the intestinal epithelial cell is caused by the interaction between polymorphonuclear leukocyte, that is neutrophils and lymphocytes, and trophocytes of Entamoeba histolytica. It degrades the DNA and mitochondria of the host and leads to apoptosis of the cell. In liver, the absence of a strong inflammatory response damages the liver cell. Similarly, in amoebic hepatic abscess, the monocyte locomotion inhibitory factor is responsible for delayed inflammation. Three main virulence protein factors, that is galectin, cysteine proteinase, and amoeba pores. These galectin, cysteinase proteinase, and amoeba pores are involved in pathogenesis. So these are the three main virulence factor which is involved. It can spread to liver, lungs, as well as brain. Clinical features of amoebiasis. Amoebic dysentery is the third most common parasitic infection and a leading cause of mortality and morbidity worldwide, mainly found in migrants and travelers. So it was not described until 1875 when Fedor Losch isolated the organism from the stool of a farmer in Russia. Infection is common in developing countries due to lack of personal hygiene and poor water supply, and is often misdiagnosed due to the unavailability of laboratory facilities. According to WHO, the mortality rate is over 50,000 to 1 lakh deaths related to amoebiasis worldwide every year. Nutritional status is probably the main cause of the increased mortality rate. Patients with acute amoebic dysentery range from 2.2% to 16%, causing invasive intestinal amoebiasis. An outbreak occurred in 3 hostels. So, by the food handlers in early 20th century in Chicago. The waterborne outbreak occurred in 1934 and 1950 when there was a direct contamination of drinking water with fecal contaminated wastewater. And last is sexual transmission also be reported as cause of an outbreak. So clinical manifestation of amoebiasis, almost 80% of the entamoeba histolytica infection is asymptomatic and can persist in the colon for a long period of time, causing a risk of infection to others. After 2 to 4 weeks of infection, symptoms in intestinal amoebiasis normally occur, causing profuse diarrhea, cramps, mild fever, abdominal pain, etc. Complications, sometimes intestinal amoebiasis can be chronic, causing vague gastrointestinal distress, blood and mucus diarrhea, severe ulceration of colonic mucosal surfaces, and obstruction of the intestine due to trophocyte masses can be seen rarely. Symptoms can vary according to the host derived factor and stage of the parasite. Invasive trophozoite infection causes amoebic dysentery, a severe form of amoebiasis that causes diarrhea, stomach pain, weight loss, fever, and chills. So, amoebic dysentery is the development of amoebic abscesses in intestinal tissue when trophozoite invade the intestinal wall and disseminates via the bloodstream. Most commonly infected extraintestinal organ infections are liver and brain, peritonitis, pleuropulmonary abscesses, and cutaneous and genital amoebic lesions. Fever, weakness, abdominal swelling, nausea, jaundice, cough, and upper right quadrant pain are the symptoms that may persist for several weeks of infection. Diagnosis. So initially, diagnosis of cyst and trophozoite is done by microscopic examination of stool samples in developing and developed countries. So other samples such as aspirates from the intestine, biopsy material, and mucus from rectal ulcers can be used for observation. Trophozoite identification can also be done by saline wet mount wet mount preparation and Viethi's trichome. To detect cyst, specimens are stained with iodine. An examination of a triplicate sample must be taken for 3 days as the organism excretion can vary daily. Microscopic examination cannot differentiate Entamoeba histolytica. So, therefore, antigenic and molecular detection must be performed. Serologic testing can be done as antibodies against Entamoeba histolytica are detectable within 5 to 7 days of acute infection but is less sensitive for liver abscesses. So, abscesses in liver, pus, stool, and sera has proven useful for detecting amoebic liver. For intestinal amoebiasis diagnosis, sigmoidoscopy or colonoscopy are used in large bowel examination. For extraintestinal diagnosis, organisms are supplemented by radiography. Treatment and prevention. Asymptomatic intestinal infection can be treated with the administration of paromomycin or dilozanide furoate. These intestinal and extraintestinal infection can be treated with metronidazole or another 5-nitronidazole. These antibiotics for amoebic liver abscesses, therapeutic aspiration is recommended unless the patient shows no improvement with oral therapy. Broad spectrum of antibiotics are recommended in case of fulminating colitis or abscesses drainage. So, treatment, prevention, and control measure. Conventional drinking water treatment should be implemented, such as sedimentation, coagulation, filtration, and use of disinfectants. Heat treatment of food at 68 degrees Celsius for 5 minutes. It will kill cysts, so properly heat the before food consuming. Using human feces as fertilizer in crops. Travelers should be careful while traveling to endemic region. Cysts resist chlorine, so food and water must be heated above 68 degrees Celsius or using iodine at 200 ppm. Sanitation and food handler screening, boil and filter water. So all these preventive measures should be taken to avoid parasitic infection because parasites are widely spread in the environment and are responsible for causing diseases associated with diarrheal illness. Foodborne and waterborne parasites are mainly associated with disease and are the greatest concern in food production. Parasitic protozoan cause the greatest risk to human health and economic loss to the nation, mainly in developing. So, all these preventive measures should be taken. Next is Balantidium coli. So, Balantidium coli is a parasite belonging to the phylum Celiophora and family Balantidae. It is the only ciliate protozoan known to infect humans and is distinguished as the largest protozoan parasite residing in the human large intestine. So, mode of transmission, balantidiasis is considered a zoonotic infection, meaning it is transmitted from animals to humans. People become infected by ingesting food or water contaminated with feces that contain the cyst of Balantidium coli. The main reservoirs are pigs, but other animals and humans who carry the parasite can also spread it. Pigs and other animals can also become human carriers. Balantidiasis, it is it is considered a zoonotic infection, meaning it is transmitted via animals to humans. Epidemiology and prevalence. So the infection was first documented in 1857 by Malmsteen, who described its presence in the feces of dysenteric patients. Balantidium coli is distributed worldwide, but its prevalence is very low, meaning few people are typically infected even in areas where it exists. Habitat. The natural habitat of Balantidium coli in human is the largest intestine. Here, it may live without causing harm but can also provoke disease under certain conditions. Morphology. Balantidium coli exists in two forms, the trophozoite and the cyst. So A is trophozoite and B is cyst. The trophozoite form is the active invasive stage seen in dysenteric stool. It's large and ovoid, covered in cilia that provide motility. Covered in cilia. So distinctive features include an anterior groove, that is peristome, leading to the mouth, that is cystome, a funnel-shaped gullet, that is cytopharynx, and a posterior anal pore, that is cytophyge. The cilia around the mouth are larger. Internally, there is a large kidney-shaped macronucleus and a smaller micronucleus plus contractile and food vacuole. So the cyst formed is spherical, around 40 to 60 μm in diameter, and has a thick double layered wall. Its cytoplasm contains the same nuclei and vacuoles as the trophozoites. The cyst is the infective stage for humans and is often observed in chronic cases and carriers. Life trophozoites. So infection starts when the person eat contaminated food or water. So in small intestine, cysts undergo excitation to release trophozoite. These trophozoites travel to the large intestine and multiply. Some invade the intestinal wall, causing ulceration and dysentery, while others remain free in the lumen. In the colon, trophozoite and cyst again, cyst passed in feces can survive in the environment and infect new hosts. So, the trophozoite is actively motile and is invasive stage of the parasite that is found in dysenteric stool. It is large ovoid cell, and the motility of the trophozoite is due to the presence of cilia that is covered the entire surface of the body. At the anterior end, there is groove peristome which is leading to the mouth. It is all about morphology, cytosome, and a short funnel. Posteriorly, there is a small anal pore. The cilia around the mouth are larger. The cell has two nuclei, a large kidney-shaped macronucleus, and lining in its concavity a small micronucleus. Last is cytoplasm has one or two contractile vacuoles and several food vacuoles. About cyst, its life cycle. So here, infection begins when a person ingests cyst. When the person eats contaminated food or water containing cysts, in small intestine, cysts undergo excitation to release trophozoite. These trophozoites travel to large intestine and multiply. Some evade the intestinal wall, while others remain free in lumen. In colon, trophozoites and cysts again, cysts will pass in feces, can survive in environment and infect new hosts. Pathogenesis. In healthy individuals, Balantidia coli behaves as a harmless commensal within the intestinal lumen. Disease occurs primarily in susceptible hosts, example those malnourished or with alcohol abuse. When trophozoites invade the mucosa, causing inflammation, ulcers and abscesses reminiscent of those seen in amoebiasis. Importantly, unlike antiamoeba histolytica, Balantidium coli does not invade other organs like the liver. Sign and symptoms. So most infections are asymptomatic, but when symptoms occur, they closely mimic amoebiasis, diarrhea, or frank dysentery, abdominal pain, which cause nausea and vomiting. Ulcer caused by Balantidium are often secondarily infected by bacteria, that is potentially leading to severe outcomes such as intestinal perforation, peritonitis, and in rare cases, death. So the genital and urinary tracts may sometimes be affected, and in chronic cases, symptoms fluctuate between diarrhea and constipation. Laboratory diagnosis, stool examination. Diagnosis is mainly through stool examination, where motile trophozoite appear in diarrheic stools and cysts are observed in form stools. So these forms are distinguishable by the large size, unique nuclei, and rapid motility. Biopsy. If stool tests are negative, biopsies or ulcer scrapings may reveal the parasites in tissue. Treatment. So tetracycline is the drug of choice, which is given 500 mg 4 times daily for 10 days. Alternatives include doxycycline, metronidazole, and other nitroimidazoles for some cases. Next is Trichomonas vaginalis. So Trichomonas species are flagellate protozoan that exist only as trophozoites. They lack a cyst stage. Three species infect humans: Trichomonas vaginalis, where the focus lies, the Trichomonas hominis, and Trichomonas tenax. Its distribution, these three species, its distribution first observed by Dunne in 1836 in vaginal secretion. Trichomonas vaginalis shows widely varying prevalence from 5% in general hospital patients up to 75% in sex workers. Its habitat in female Trichomonas vaginalis inhabits the vagina and cervix as well as glands, urethra, and bladder. In male, it is found in the anterior urethra but may also affect the prostate and perpetual sac. Morphology. The trophozoid form is pear-shaped or ovoid, which measures around 10 to 30 μm long and 5 to 10 μm wide. It has 4 anterior flagella plus a fifth associated with a short undulating membrane that extends halfway down the body. The membrane is supported by a costa. A prominent exostyle runs through the body and protrudes as the posterior end. Movement is rapid and jerky, that is twitching. It is the shape of Trichomonas vaginalis. Its life cycle. So only the trophozoite is infectious, as there is no cyst stage for trichomonas. Transmission is mainly sexually. Rarely it can occur via contaminated objects. Upon entry, trophozoite colonize the urogenital tract and multiply by binary fission, causing local irritation and discharge. Transmission is direct from one person to another because the parasite does not survive long outside the host. Pathogenesis. Pathogenesis. Trichomonas vaginalis infects squamous epithelium and secretes a cocktail of enzyme and acids that lower vaginal pH and disrupt glycogen metabolism. The organism is an obligate parasite depending on close association with host tissue. Infection leads to strawberry mucosa, epithelial changes, and intracellular edema. These are hallmark features. Edema is nothing but swelling. Sign and symptoms. The infection is frequently asymptomatic, especially in men, but can cause urethritis, epididymitis, or prostatitis. Women can experience severe, offensive, yellow-green frothy discharge, pain during urination, and dysphagia. So cervical erosion is common. Rare complications include endometriosis, pyelopharyngitis, and neonatal pneumonia or conjunctivitis in infants born to infected mothers. The incubation period is around 4 days to it will last 4 weeks. Laboratory diagnosis, microscopic examination, microscopic wet mount of vaginal or urethral discharge is used to detect the characteristic motile trophozoites, often accompanied by abundant leukocyte in males. They may be seen in urine or prostatic fluid. Second is serology. ELISA technology employing monoclonal antibodies detects Trichomonas vaginalis antigen directly from vaginal smear samples. Third is molecular method. So DNA hybridization and PCR are highly sensitive and specific diagnostic methods for trichomoniasis, confirming the presence of Trichomonas vaginalis genome in clinical samples such as stool or genital discharge. Laboratory diagnosis and treatment. So molecular method is DNA probe and PCR. DNA treatment, again metronidazole and tinidazole are the drug of choice for treating trichomoniasis. Next is Giardia lamblia.
Madam,
Hello, yes sir.
madam, up to what time you are going to take, madam?
Around 8:30 sir.
8:30. Okay, madam. You can continue.
All right sir. Next is Giardia lamblia. So Giardia, it is a foodborne disease. So protozoan causing malabsorption syndrome and diarrhea. It is very common in children and low sanitization regions. Here is a diagrammatic representation of Giardia. So the main source of contamination of Giardia is the feces of pets, livestock, and wild animals, and is commonly transmitted through the fecal oral route. First, we'll go with characteristics of Giardia. It has flagellated protists, tumbling motility, diplomonads, that is two bodies having individual nuclei, mitochondria replaced by mitosome, metabolizes by anaerobic glycolysis. The life cycle completes at two stages, a reproductive trophozoid and a cyst stage. It reproduces asexually. The reproductive trophozoid is around 9 to 21 μm long and cysts are around 9 to 12 μm long. Giardia cysts are susceptible to disinfectants like chlorine, iodine, and chlorine dioxide. So, the main source of contamination is through fecal route. Giardia cysts are also found in sewage, effluent, and shallow spring water, as well as cysts, and are very infectious and can remain for a long period in a cool and damp place. Giardia is resistant to chlorine and can survive some common water treatment, creating a great risk to water supplies. They can be removed by filtration process, but Cryptosporidium cannot be removed by filtration because of their smaller size. So its morphology, it is flagellated protozoan with two nuclei. As you can see here, two nuclei is there. Trophozoites around 9 to 21 μm, cyst is around 9 to 12 μm, and it is chlorine resistant. Transmission through fecal oral route via animals, water, and sewage. Its life cycle. So through ingestion of cyst, the life cycle of Giardia completes in two stages. First, the cyst that initiate infection, and the second, vegetative trophozoid, which replicates and causes diseases. So the cysts are environmentally resistant, especially at low temperature, because of their outer filamentous layer made of N-acetyl galactosamine and three proteins, namely CWPs, that is 1 to 3, which allows them to survive for a long period of time. So about 10 to 100 cysts are capable of causing infection when it passes through the stomach acid and reaches the duodenum. So after entering the duodenum, true trophozoites are formed from each cyst and colonize in the small intestine by absorbing essential nutrition and causing illness to the host. Giardia reproduces asexually, therefore trophozoite replicate by the mode of binary fission and are tetraploid, that is contain 4 sets of chromosome. So the above cycle is followed by excitation when the cyst pass through the stomach's acidic environment to the duodenum and in small intestine, and then excitation takes place. So in response to the trophozoid, T lymphocytes are activated by the body's immune system, which disrupts the epithelial microvilli and shortens them. So when it shortens them, it also disturbs the epithelial tight junctions that lead to an increase in permeability and diarrhea. The cyst also goes through bile salt and cholesterol starvation, starting its replication and getting divided into two trophozoites and infecting the other host. It is also evident that the host acquires a resistance to the infection from an effective immune response. So here the organism is reported to have adapted to the different intestinal environments according to their antigenic variation. Although the organism does not produce any toxin or virulence factors, some organisms with variant-specific protein, that is VSPs, build resistance to antibodies. So these are the steps of infection. Pathogenesis is divided into two stages, as I mentioned earlier. So first is the cyst and the second is vegetative trophozoid stage. Cyst and trophozoid stage. Clinical features. It is an infective dose. It includes around 10 to 100 cysts, and symptoms are diarrhea, bloating, malabsorption, weight loss. So the protozoan parasites Giardia causes approximately 4 to 20 cases of outbreak per year, and most are reported to be caused by the fecal oral transmission route. Giardia is reported as the 11th position out of 24 foodborne parasites according to the Food and Agriculture Organization and WHO. The epidemiology of Giardia is more prevalent in developing countries and is also associated with environmental and climatic conditions. Children under 5 years are at high risk as compared to adults, and about 60 to 80% of infections are asymptomatic. The illness is usually asymptomatic and leads to stunning growth in children at an early stage of growth. It was reported to be a waterborne outbreak as the excretion dose is higher than the infective dose per day. An infected person excreted up to 110 to 100 cysts per day, but the infective dose required to cause infection at a time. The possible rate of contamination is high in developing countries where people consume tap water, shallow wells, and surface water. These water sources are rich in zoonotic parasite, and when consumed without purification, it will lead to human infection. The giardiasis infection usually peaks in the late summer as the consumption of water is high at this time, and most children are affected in daycare centers. An outbreak that occurred was from the consumption of home canned salmon infecting 60 employees at school, but the outbreak was not reported as giardiasis as the transmission was asymptomatic carriers of giardia. Diagnosis. The giardia cyst in feces sample can be identified by the use of microscope, but this traditional method lacks specificity and sensitivity. Immunoassay methods such as enzymatic immunoassay, EIAs, and immunochromatographic tests are also used, but they also lack specificity due to the cross reaction between antibodies. The most widely used method by the US Environmental Protection Agency is the separation of cysts by an immunomagnetic method. After separation, the cysts are visualized under microscope by staining them with fluorescent dye and differential interference contrast, that is DIC. The last is PCR-based method, which offers improved specificity and sensitivity as compared to other detection method. Treatment and prevention, these are the choices of drugs. Prevention, municipal water treatment, that is filtration plus chlorination. Heat treatment around 55 degrees Celsius for 5 minutes. And most important is hygiene. Next is Leishmania. So the species of Leishmania donovani was reported simultaneously by Leishmania from London and Donovan from Madras in 1903. Hence the name is Leishmania donovani. So Leishmania donovani causes a malaria-like fever, oriental disease in the man called Kala-Azar dum-dum fever or black fever. Leishmania donovani, the causative organism of visceral leishmaniasis, Kala-Azar in Africa, the Middle East. So it is endemic in Asia, Africa, the Americas, and the Mediterranean region. Its vector is sandfly. Morphology. The parasite occurs in two forms or stages, that is leishmanial or amastigote, so which alternate between a vertebrate man and an invertebrate, that is sandfly host. Morphology. So these leishmanial or Leishmania donovani, the parasite occurs in two forms or stage, leishmanial or amastigote and leptomonad or promastigote, which alternate between a vertebrate and invertebrate. So this stage occurs intracellularly in blood cells or reticuloendothelial cells of the vertebrate host or man. It is microscopic, rounded or oval in shape, measuring around 2 to 4 micrometer in length. There is no free flagellum, so it is greatly reduced, fibril-like, and lies embedded in the cytoplasm. A flagellar stage of amastigote is known as LD bodies. The nucleus is central or eccentric. This eccentric, the cell membrane is delicate and can be demonstrated only in a fresh specimen. Kintoplast is rod-shaped or dot-like and lies at the right angle to the nucleus. The exoneme is a delicate filament extending from the kintoplast kintoplast to the margin of the body. It represents the foot of the flagellum. They are stained well with gypsa or ricin. In a gypsa stained preparation, the cytoplasm surrounded by a limiting membrane appears pale blue. The nucleus relatively is larger and stained red. The kintoplast stain is deep red. Amastigote divides by binary fission at 37 degrees Celsius. So it is created with biorender. So life cycle, sandfly injects promastigotes, then it macrophages will happen, and then amastigotes, then it will lead to spread in spleen, liver, and last is bone marrow. So leishmania is also a digenetic parasite that requires two hosts for completion of its life cycle. The primary host is a vertebrate or man, in which the parasite feeds and multiply asexually. And second is vector invertebrate or blood-sucking insect or sandfly. First human stage, sandfly stage, belonging to the genus Phlebotomus. So some mammals like dogs, jackals, gerbiles, and squirrels also serve as a reservoir host in which the parasite does not undergo any change but simply waits for its introduction to the human host. So life cycle in men, the parasite has two stages in its life cycle. Amastigote form occurs in humans and mammals. Promastigote form occurs in sandfly. So, Leishmania donovani is transmitted to humans or other vertebrates by the bite of blood-sucking sandfly, that is Phlebotomus argentipes. The parasite introduced by sandfly into the human body are the promastigote form. Some of the promastigote entering the blood circulation directly become destroyed, while those entering the reticuloendothelial system, that is liver, spleen, bone marrow, and lymph node, change into amastigote or leishmanial forms. So the amastigotes multiply by simple binary fission inside the reticuloendothelial system to form a large number of amastigotes. When the number of parasite reaches around 50 to 200 or even more, the host cell will rupture. So the liberated parasites are taken up by new host cell and the multiplication cycle is repeated so that the reticuloendothelial system becomes progressively infected. Some of the free amastigotes are phagocytosed by the neutrophils and monocytosed in the bloodstream. They are heavily parasite cells wandered through the general blood circulation, which leads to general infection. Life cycle in sandfly. So when the sandfly sucks the blood of an infected person, it obtains free amastigote as well as the parasites, neutrophils, and monocytes along with the blood meal. So the parasite begins a process of transformation and the amastigotes change to procyclic promastigotes and then the metacyclic promastigotes in the midgut of the sandfly. So these promastigotes multiply by longitudinal binary fission and produce large number of promastigotes which completely filling the lumen of the gut. In 6 to 9 days, the number of parasites become enormous and heavily spread into the pharynx and buccal cavity. The salivary glands are not infected here. Transmission into new host occurs when such a heavily infected sandfly bites the host. Its clinical features: fever, weight loss, dark skin, hepatosplenomegaly, anemia, all this will include under clinical features. Diagnosis is through microscopy, bone marrow or splenic aspirate, serologic through ELISA, and that last is molecular method, that is PCR. Treatment and prevention. Treatment, these are the choice of antibiotics. Prevention, vector control, you can take nets or insecticides. Early diagnosis, and it is still yet not discover any vaccine. Next is malaria, plasmodium species. Introduction, it is caused by Plasmodium falciparum, Plasmodium vivax, Plasmodium ovale, Plasmodium malariae, Plasmodium lacei. Its vector is female Anopheles mosquito. Transmission of malaria, it is through mosquito bites, blood transmission, and congenital. Life cycle of malaria. So life cycle, it includes nature that alternates between the female Anopheles mosquitoes and humans. So there are two types of life cycle: human phase and mosquito phase. In human phase, first it occurs in liver, then lead to blood. It includes merozoites and gametocytes. And in mosquito phase, it will occur in salivary gland, that is sporozoites. So human, the parasite initially proliferates in the liver cell, and then the red blood cells of humans. Within the red blood cells, plasmodium develops into schizons, which ruptures the RBCs to invade other neighboring RBCs. The schizons then develop into merozoites. This stage of the plasmodium life cycle is responsible for causing symptoms of malaria in humans. Some parasites differentiate to form sexual erythrocyte, which is known as gametocytes. Eventually, the gametocyte forms parasites are ingested by female Anopheles mosquito vector during blood meal, which signals the start of the second life cycle. These are the graphical representation, diagrammatic representation of life cycle of malaria in liver. So first infection will occur, then sporozoites form. Merozoites will occur in liver cell, and then it will release. Red blood cell get infected, it will form ring structure, then trophozoite, then schizon, and lastly, like this, infection occurs. Clinical features: high fever, chills, anemia, jaundice, cerebral malaria, all this will include in clinical features. Diagnosis: malaria is one of the deadliest disease and requires a thorough and immediate response. Usually, diagnostic method involve infection of the parasite itself. So, it will include thick and thin blood smear, RDT, as well as molecular method, that is PCR. Treatment and prevention. These are the choice of treatment: drug, prevention, mosquito control, chemoprophylaxis, and vaccines for malaria. Next is three types of spores: cryptosporidium, cyclosporidium, microsporidium. So all this overview, these are the spore forming intestinal protozoan parasite. It will mainly infect the digestive tract, causing diarrheal illness. It will affect both immunocompetent as well immunocompromised individual with more severe disease. Patients having underlying health issues, that is HIV and AIDS, and malnourished patient. Transmission is through contaminated food, water, or person-to-person spread. It is resistant to standard chlorination and can persist even in the environment. Cryptosporidium. So its etiology, it is mainly C. hominis, and that is Cryptosporidium hominis and Cryptosporidium parvum. It causes cryptosporidiosis, leading up to cause childhood diarrhea. Infection begins when oocytes are ingested. It will release sporozoin and then invade enterocytes in small intestine. Autoinfection, some oocytes remain in host, which will lead to persistent infection. Clinical features, incubation period is around 2 to 14 days. Symptoms include profuse watery diarrhea, abdominal cramp, nausea, anorexia, fatigue, vomiting in children. Severe cases include malabsorption, weight loss, dehydration, chronic and fatal in immunocompromised patients. Diagnosis is through microscopy, ELISA, molecular method PCR, and histology. Treatment, drug immunocomputer often self-limited to rehydration. All this choice of drugs will be given according to the age of patient. HIV patient, combination therapy will be given, and supportive therapy, oral rehydration, and nutritional support. Sir, can we continue in next class? Or should I continue? Is it fine?
Madam, as per your strength,
Yes, sir. No, I am comfortable, sir. If you don't have any other class or work, I can continue.
so recently you are suffering with viral fever.
Yes, sir.
So just see whether you have a capacity, you can continue. Otherwise, you can next class also.
We can continue then in next class. So in detail, I can explain all this parasites. No, it will
Yeah,
be okay. All right, sir. Then we can continue in next class.
yeah, please, one. Yeah.
All right, sir. Thank you, sir.
Thank you. Thank you so much.
Sure. Thank you.